Okay, so in this video, we're going to continue our discussion of the consequences of meiosis. We have two big things to finish up on here. The first is introducing the concept of recombination. Second is to, as I said, we've been talking about kind of fake simplified meiosis. So now we're going to introduce back the extra step that we've been avoiding or ignoring. So we're going to go to real meiosis, and then finally we just put these two things together to show how recombination works in real meiosis. Okay, so we discussed how meiosis is a way that mutations get reshuffled, so that we can start with haploid cells, each of which have one mutation, and then after fusion and meiosis, we can end up with meiotic products that have different combinations. However, so far we've only talked about when these mutations are on different chromosomes, on the orange set and on the green set. But what happens if the two mutations are on the same chromosome? So now if we have fusion, if we only shuffle the chromosomes as we've discussed, we're still only going to end up with either this orange chromosome or this orange chromosome. And so we'll either have mutation 1 or 2, but not neither and not both. So the concept of recombination is going to show us how we can reshuffle mutations that are on the same chromosome. So first, remember that during meiosis, that homologous chromosomes, the thick and the thin, are going to line up together on the metaphase plate, as we show here. And this gives the opportunity, this physical proximity gives the opportunity for these chromosomes to recombine. So I'll show what I mean here. So if we zoom in on the orange chromosomes, so if we have our mutation 1 here and our mutation 2 here on the other homologous chromosome, what can occur is that we can have a breakage and rejoining event where the top of this chromosome becomes joined to the bottom of this one, and the top of this one becomes joined to the bottom of this one. So now let's follow through what happens when these get drawn. So here are our microtubules attaching and pulling our chromosomes to the opposite poles. And so what we see is that we end up with these hybrid chromosomes, these chromosomes that are a combination of our two starting homologous chromosomes. And we see we have one copy that has neither mutation, and we have one copy that has two mutations. So what we've shown is that the process of reshuffling happens not only between mutations or variation on different chromosomes, but can happen between mutations or variations on the same chromosome by this physical process of breaking and rejoining called recombination. So let's do this with a little bit of sequence so we get accustomed to it. So I've drawn this a little differently than we're accustomed to. So take a moment to orient yourself. So here you can see each one of these here is a base pair, A with T, G with C, C with G, and so forth. And what you'll see here is that it's almost exactly the same, AT, GC, CG, CG, AT, and so forth. But then we can imagine a mutation or two mutations where we have two sites along these chromosomes that differ between chromosome 2 and chromosome 1. Okay, so I've shown a very small number of base pairs here. Remember that if this is the human genome, then this could be 100 million base pairs. So this is not to scale, but it'll allow us to understand the concept. So we have a double-stranded DNA and a double-stranded DNA. And then during recombination, what will happen is at a given site that this chromosome will get attached to this one, and this one will get attached to that one. And so if we follow this through, let's use red to mean our left combination and blue our right combination. So if we would start out with red, red, and blue, blue, but then after recombination, now we're going to have red with blue. That's our chromosome on the right here. And we're going to have blue with red. That's our chromosome on the left. So we can see that physically at the level of sequence, this is how recombination is going to reshuffle our variation. And so then we can ask a question about, for this meiosis depicted, what's the chance that a chromosome will be formed that will have both mutations, 1 and 2? Think about it for a moment. Okay, so here's something new that we can introduce. So we get about one recombination, or maybe a couple of recombinations per chromosome length, the whole chromosome length per meiosis. And so what that means is that these mutations are at fairly close sites together, and so we're only going to form a hybrid chromosome with 1 and 2 if the recombination occurs specifically in this region. Because if the recombination occurs in this region, well, let's trace out what that would mean. That would mean we'd have this whole top of the thick chromosome, so we'd have mutation 1, and then the bottom here, so we would not have mutation 2. And then on the other case, we would have this whole top of the thin chromosome, which would be mutation 2, and then the thick bottom, so not mutation 1. So we're only going to get reshuffling if we have a recombination right in between in this short region. So when you look at two mutations, if they're near each other on the chromosome, then it's unlikely that they are going to be recombined together, and more likely that they will stay as they are. And we'll learn how to formally talk about this in the next video on recombination rate. Okay, so next up is to go into real meiosis. So as you'll remember, we've been dealing with this thing I've called fake meiosis, which is slightly simplified. And in that case, what we have is we have fusion of two haploids, giving rise to a diploid, and then this diploid, in the diploid, without any other changes, we have lining up of the homologous chromosomes and separation. So in real meiosis, there's one extra step. And so what happens in that extra step, well, in fact, there are two extra steps. We have a DNA duplication step, and that requires a second division. So the first thing that happens is that each of our chromosomes, all of our skinny chromosomes that came from one parent, and all of our thick chromosomes that came from the other parent, undergo duplication. So now we have twice as much DNA as we would in a diploid. It's like when a diploid undergoes mitosis, and so first has to duplicate. And so now, as before, now all the green chromosomes, that is all the homologous pair, are going to line up. All the blue are going to line up together, and all the orange are going to line up together. But now, instead of having two double-stranded DNAs in each group, we have 4. See? 2 of the skinny and 2 of the thick. And so now we're eventually going to get to haploid cells, but now we have 4 copies. So we're actually going to have to undergo 2 rounds of division. So in the first round of division, what's going to happen is that these 2 attachment points, these centromeres to the microtubule, they're going to get dragged to the right, and these 2 are going to get dragged to the left. These 2 are going to get dragged to the left. These 2 are going to get dragged to the right. And so then what we end up with is, ignore for now the apparatus for division, but if we just look at the chromosomes, what we see is we have the homologous chromosomes, or rather the duplicated chromosomes, here, here, and here, and here, here, and here. Just these in the right cell and these in the left cell. So this is a full cell division that gives us rise now to copies with cells with 2 copies each, 2 double-stranded DNAs for each homologous chromosome type. And so now, as you see depicted here, these are going to line up again, and they'll line up for another round of metaphase, another round of division. And now these are going to pull apart, and we end up with 4 cells. So you can see this has outcomes very similar to meiosis, or to fake meiosis, in that we have shuffling of skinny and thick. And the major point here is we end up with 4 cells because we have this round of DNA duplication, which requires a second division. So, 3 things to point out here. We have DNA duplication, 2 divisions, leading to 4 meiotic products per meiosis. And so now it's worth pointing out what recombination looks like in this case. So before, remember we just did one copy each of each homologous chromosome. Now we have two copies each. We have two full skinny double-stranded DNAs and two full thick double-stranded DNAs. And so just as before, we can have recombination. So here we can show a recombination between this thick and this skinny, giving rise to these hybrid chromosomes. But we can also have recombination between other pairs. So physically, we've represented these things as all being apart from each other, but they're all close enough that we can have recombination between any pair of double-stranded DNAs. And so we can get a recombination where this thick part is now attached to this thin part. And so then when we sort that out, we would get one chromosome that has a big chunk of thick and then a whole lot of thin, one that has a small chunk of thick and a whole lot of thin, and vice versa. So in this video, we introduced the concept of recombination. We talked about real meiosis, adding in the extra duplication and the extra division. And we talked about what recombination would look like on real meiosis.